which codes for the constant region of the antibody. The mu gene codes for the protein that makes the IgM constant region and is the first of the different types of antibody constant regions that are expressed on B cells. Once a cell has successfully completed VDJ rearrangement on the heavy chains and bound it to the mu constant gene, it's considered a large pre-B cell. At this point, it's time to test out if the heavy chain is functional, by seeing if it can bind to a surrogate light chain and get expressed on the cell surface. The surrogate light chain is made of two proteins, V pre-B and lambda 5, and it's basically something for the heavy chain to practice with until the real light chain is eventually made. The surrogate light chain and heavy chain are joined together and placed in a vesicle, and then that vesicle is sent up to the cell surface. At the cell surface, the surrogate light chain and heavy chain are placed near two additional chains, immunoglobulin alpha and immunoglobulin beta, which are called side chains. If the surrogate light chain and heavy chain successfully make a functional heavy chain on the surface of the large pre-B cell, these side chains send down a signal to the nucleus. The cell starts to rapidly proliferate, and each daughter cell is called a small pre-B cell. The small pre-B cells now begin rearranging the light chain, which means that the pre-B cells will all have the same heavy chain, but each will have its own distinct light chain. And there are actually two types of light chains, a kappa light chain and a lambda light chain. The kappa light chain starts rearranging first, and it's an iterative process. In other words, initially a pre-B cell might create a kappa light chain that doesn't bind well to the heavy chain. If that happens, the cell will either get a chance to iterate and try a different arrangement, or in some cases the cell might simply get killed off. Not allowing B cell receptors to be self-reactive is super important, because if these faulty or self-reactive B cells are released, then they might simply not work and they might attack self-tissue and cause disease. To identify the self-reactive B cells, there's an autoimmune regulator gene, called AIR for short. AIR is expressed in primary lymphoid organs and allows them to express antigens that are normally found all over the body, like the brain, ovaries, and pancreas. It allows these primary lymphoid organs to serve as a microcosm of the entire body, kind of like how the Epcot Center theme park in Florida is a microcosm of the world. So that allows the body to test out these newly formed B cell receptors in a safe environment. If a small pre-B cell binds strongly to a self-antigen, then it dies swiftly and silently by apoptosis. Whereas if a small pre-B cell binds with only intermediate strength to the self-antigen, then the small pre-B cell will try to rearrange its light chain in the hopes that it'll reduce the strength of its binding to a self-antigen. Only small pre-B cells that don't recognize self-antigens at all develop into immature B cells. Now, if the B cell does have a faulty B cell receptor or a self-reactive B cell receptor, then the pre-B cell tries again by splicing out more DNA segments from its light chain to generate a new V and J rearrangement. In fact, the cell will keep trying to do this over and over, doing more and more rearrangements, until it's gone through all of the V and J segments on the first chromosome. And then it starts to splice out DNA segments on the second chromosome. Once all of the V or J segment rearrangements on the kappa chain of both chromosomes have been tried, the cell will start trying to rearrange the lambda chain, and it'll keep iterating with the lambda chain as well, until it runs out of genetic material. Ultimately, if there's a rearrangement where the resulting light chain binds to the heavy chain, and the resulting B cell receptor isn't self-reactive, then and only then does the cell become an immature B cell. Now, to get into the blood, an immature B cell has to signal to the bone marrow that it has completed its development. Now, remember that the B cell already has a B cell receptor with a mu heavy chain constant region, which made it an IgM antibody. But the B cell now undergoes a process of alternative splicing, which is where some of the B cell receptors continue to use the IgM constant region, whereas some start using the IgD constant region. But all of the antibodies, both IgM and IgD, still have the same antigen binding specificity. The presence of the IgD constant region on some of the B cell receptors is a bit like a driver's license. It signals to the bone marrow that the B cell is a fully rearranged immature B cell that should be allowed out on the open road. Alright, as a quick recap. B-cell development includes rearrangement of the heavy chain, rearrangement of the light chain, and making sure that the B-cell receptor isn't faulty or self-reactive. Heavy chain rearrangement means combining the DJ segments and then the VDJ segments. And then the VDJ segment is joined to an IgM heavy chain, and then it's paired with a surrogate light chain before the cell proliferates. Each daughter cell rearranges its light chain by combining the VJ segments, first with the kappa chain genes and the lambda chain genes. If the B-cell receptor isn't faulty or self-reactive, then it'll express the IgD constant region, which will allow it to be released into the periphery as an B-cell. Despite being surrounded by harmful microorganisms, toxins, and the threat of our own cells turning using the process of alternative splicing to allow for some of the IgM to be secreted. The immune system is highly specific for each invader, and that's because the cells of the adaptive immune response have unique receptors that can differentiate friendly bacteria from potentially deadly pathogens from their unique parts, called antigens. The key cells of the adaptive immune response are the lymphocytes, the B and T cells. B cells develop in the bone marrow, where they undergo a process called VDJ rearrangement to generate a massively diverse set of B cell receptors. The B cell receptor is essentially an antibody, except that it has a transmembrane part that goes through the membrane, attaching the receptor to the surface of the B cell. The B-cell receptor has 2 heavy chains and 2 light chains. And the region or fragment of the B-cell receptor that binds the antigen is called the fragment antigen binding, or FAB region. The FAB region is where the ends of the heavy and light chains meet, and there are 2 FAB fragments on each B-cell receptor. The remainder of the heavy chain makes up the constant region, or constant fragment region, also called FC. The 2 heavy chains are linked to each other by disulfide bonds, and each heavy chain is also linked to a light chain by a disulfide bond. Each B-cell receptor has 2 identical heavy and light chains, resulting in 2 identical antigen binding sites. As the B-cell develops into a plasma cell, the B-cell receptor gets secreted as an antibody with the exact same antigen specificity. However, the heavy chain actually changes as the B-cell develops. There are 5 major types of heavy chains, which encode the isotypes or classes of immunoglobulins: IgM, IgD, IgG, IgA, and IgE. These 5 are encoded by heavy chain genes, which are referred to by the Greek letters mu, delta, gamma, alpha, and epsilon. When a B-cell is first developing, it initially expresses the mu heavy chain, and as a result, all of the B-cell receptors are IgMs that are on the cell surface. When the B-cell finishes developing, it undergoes a process called alternative splicing. Alternative splicing is a process by which the cell expresses the heavy chain exons for both mu and delta, allowing for both IgM and IgD to be simultaneously expressed on the surface. At this point, the B-cell is mature, but still naive. Having IgD on the B-cell surface is like a young adult with a driver's license.
Smile, please.
They're able to go out and explore the world. In the B cell's case, that means all of the body's lymphatic system. But these cells haven't been exposed to much, and don't know how they'll react to foreign antigens. Once the B cell starts to explore the body's lymphatic system, they travel from lymph node to lymph node in search of antigens. Lymph nodes are scattered throughout your body, and each one is a highly organized structure where millions of B cells, T cells, antigens, and antigen-presenting cells pass through every day, kind of like a busy airport. Now, both B and T cells, as well as antigens and antigen-presenting cells, all need to enter the lymph node. There are two ways to do this, depending on if the cells are entering from the lymphatics or the blood. If the cell is already in the lymph, then it can enter the lymph node through the afferent lymphatic vessel. If the cell is in the blood, then it can use the high endothelial venules, or HEVs. HEVs are special endothelial cells found at post-capillary sites of the lymphoid organs. These endothelial cells are more plump than the average flat endothelial cell, and they also express adhesion molecules like L-selectin. When lymphocytes pass through the HEVs, they can bind to and wiggle through the endothelial cells and slip right into the lymph node. When B cells and T cells get into the lymph nodes, they first enter the paracortical region. The T cells stay there, while the B cells migrate to the neighboring cortical region of the lymph node, where they form the primary lymphoid follicles. If a B cell gets activated, it starts replicating within a follicle, and it forms a germinal center. A follicle with a germinal center is called a secondary lymphoid follicle. Various antigens enter the lymph node through the afferent lymphatic vessel, and they percolate through the paracortex and through to the follicle. It looks kind of like a game of Plinko, where the antigens get to interact with a lot of B cells in the follicle. B cells, unlike T cells, can recognize a wide variety of antigens, including peptides, carbohydrates, and lipids in their native form, meaning that they don't require antigen-presenting cells to process or present the antigen. In order for the B cell to be activated, the antigen has to cross-link the B cell receptors. When two B cell receptors get cross-linked, their intracellular chains, the side chains Ig alpha and Ig beta, and CD19 all cluster together. Each of these chains have something called an immune receptor tyrosine-based activation motif, or ITAM for short. The ITAM is a conserved sequence of amino acids that includes two tyrosine amino acids. The tyrosines and the space between the two tyrosines is super important, because both tyrosines need to be phosphorylated for the cell to get activated. These two tyrosines within the ITAM region of the protein get phosphorylated by three protein tyrosine kinases, called BLK, FYN, and LYN. Think of them as a friendly little trio of phosphorylators. Once the two tyrosines get phosphorylated, there's a chain of events that ends with the activation of the major transcription factors, NF-kappa B and NF-AT. These transcription factors turn on the expression that result in more cytokines, like interleukin-1, interleukin-2, interleukin-6, and tumor necrosis factor alpha. They also cause upregulation of anti-apoptotic cell surface markers, like BCL2, as well as the proliferation and differentiation of B cells. An entirely different, alternative way for a B cell to be stimulated is using the molecule CD21, which is also called CR2 because it was the second receptor identified for one of the complement fragments, C3d. In the complement pathway, a variety of complement fragments are produced, many of which can bind non-specifically to antigens. C3d can bind to an antigen and